When one of the many different types of barriers of our body are breached, for example, we have a cut in our skin and our pathogen makes its way into the tissue of our body, infection begins. And as soon as infection begins, the innate immune system uses non-specific defense mechanisms, such as the process of inflammation. And what inflammation does is it ultimately localizes our infection to that specific region. It calls upon macrophages and leukocytes that essentially engulf the pathogens as well as our infected cells. Now, while this process is taking place and while the innate immune system is preventing the spreading of this pathogen, of this infection, the adaptive immune system, also known as the acquired immune system, the other part of our immune system, begins to mobilize itself. It begins to gear up to basically help fight that infection. Now, we know the innate immune system begins to act immediately following infection, but our adaptive immune system actually takes several days before it is actually fully active and fully functional. Now, unlike the innate immune system, which uses non-specific mechanisms, the adaptive immune system actually uses specific defense mechanisms because it uses antigens and antibodies, as we'll see in just a moment. Now, the innate immune system cannot actually learn from infections, but as we'll see in just a moment, the adaptive immune system can actually learn from the different types of infections and pathogens that infect the body. Now, the adaptive immune system can be subdivided into two categories, into two divisions. We have the antibody-mediated immunity, or the humoral immunity, and we also have the cell-mediated immunity. And these two systems work together to basically carry out that same exact function to fight off our pathogens that infected our body. So let's begin with the antibody-mediated immunity. Now, the leukocytes involved, the white blood cells involved in the antibody-mediated immunity are the b-lymphocytes. Now, b-lymphocytes not only form in the bone marrow, but they also mature in our bone marrow. And once they are released into our plasma, into our lymph, they travel and are stored within the lymph nodes of our body. Now, when infection takes place, the b-lymphocytes are released from the lymph node and they travel through our lymph system, through our blood system, and they travel inside our tissues. Now, before we actually discuss these b-lymphocytes, let's backtrack and go back to the process of inflammation. So, when the innate immune system carries out inflammation, it basically calls upon macrophages. So, this is our macrophage. Now, what the macrophage does is it engulfs and breaks down these different types of pathogens. And what it also does, it takes the antigens of these pathogens and places the antigens on the surface, on the membrane of the macrophage. The question is, what for? Well, because as it turns out, the b-lymphocytes as well as the t-lymphocytes, as we'll see in just a moment, contain these receptors on the membrane of the lymphocyte. So the b-lymphocytes contains the B-cell receptors. And when the b-lymphocyte approaches our macrophage, if the B-cell receptor complements this antigen, we can have the binding process take place. They will form a complex, and that will ultimately call upon a cell involved in the cell-mediated immunity known as the helper T cell. And what the helper T cell does is it releases a special type of chemical, or special types of chemicals, which basically help the B lymphocyte undergo mitosis and differentiate into two different types of cells. So the B lymphocytes form plasma cells and memory B cells. And the memory B cells are the same cells that we spoke about earlier that are involved in the process of learning. So plasma cells are those specialized cells that are factories for antibodies. So they have extensive endoplasmic reticulum that essentially forms many antibodies. And these antibodies that are formed by the plasma cell are specific, and they complement the antigen that the B lymphocyte initially was bound to. And because we have many different types of plasma cells, we have many different types of antibodies that are produced for each one of these different antigens that came from each one of these different types of pathogens. Now, what exactly is the purpose of these antibodies, and what are these antibodies in the first place? Well, antibodies are specialized proteins. They are also known as immunoglobulins. Immune means immunity, and globulins mean protein. So these antibodies circulate along our blood system and our lymph system until they basically bind to the specific antigen that is found on another pathogen, or let's say some type of bacterial cell. Now, once our antibody binds onto that antigen, it can do two things. It can either call upon other leukocytes, for example macrophages, to come and engulf that pathogen, or it can begin to aggregate and form this very large insoluble complex in a process known as agglutination. So antibodies are very important, and they're involved in the adaptive immune system. And that's exactly why we call it an adaptive immune system, because we have these other types of cells known as memory B cells, and we also have actually memory T cells, as we'll see in a moment. And what these memory cells do is they store a copy of that antibody on the membrane of that memory cell. And so these memory B cells swim around and circulate our blood system and our lymph system. And if that same type of pathogen ever reinfects our body, these memory B cells, as well as memory T cells of the cell-mediated immunity, can actually recognize these pathogens immediately and begin a very quick and effective response that can ultimately kill off that reinfecting pathogen. So these are the two types of cells that come from our B lymphocyte, but these B lymphocytes must associate with the helper T cells to actually become these two different types of cells. So these two different divisions, although they are divisions, they have to work together to actually carry out this process of defending and protecting our body from pathogens. Now, the humoral immunity system is actually very effective against bacterial cells, different types of parasites, fungi, viruses, as well as toxins that enter our tissue of our body. Now, let's move on to our cell-mediated immunity. Now, the cell-mediated immunity also involves a special type of leukocyte known as the t-lymphocyte. So, B lymphocytes are the humoral immunity, but t-lymphocytes are part of our cell-mediated immunity. Now, T-lymphocytes also are formed inside the bone marrow, but T-lymphocytes mature inside the thymus, which is basically a gland found in this region of our body. And what the thymus does is it basically checks, it tests these premature T lymphocytes, and it ensures that the T lymphocytes don't actually attack our own cells of the body. And that's an important process, because if the T lymphocytes do attack the cells of our body, what the thymus does is it ultimately destroys these T lymphocytes. Now, one very important type of T lymphocyte that we spoke about earlier is the helper T cell. So once infection begins, some of these T lymphocytes differentiate into our helper T cells. Now, what exactly is the purpose of helper T cells? Well, helper T cells help differentiate other cells of our immune system, and they also release special types of chemicals known as interferons as well as interleukins. And these chemicals are involved in differentiating our cells as well as in promoting the process that defends our body from pathogens. For example, we know that helper T cells help differentiate the B lymphocytes into these plasma cells and memory B cells. Now, memory B cells help our immune system with learning, and the plasma cells produce our antibodies that basically float and circulate around our blood system. Now, what the helper T cells also do is they also help differentiate T lymphocytes into a special type of soldier cell we call the killer T cell or the cytotoxic T cell. Now, these cytotoxic T cells basically travel to the infected area, and they can use the antibody found on their cell membrane and bind to special antigen portions, specific antigen portions found on the infected cell or pathogen cell, and they can basically release these very powerful proteins, proteolytic proteins, that essentially drill holes inside those infected cells, killing those cells. And these powerful proteins are known as perforins. So basically, the T lymphocyte, with the help of the helper T cell, differentiates into these soldier cytotoxic T cells that contain these receptors. And when these receptors locate the specific type of antigen found on the infected cell, they bind, they form a complex, and once they form a complex, they begin to release these powerful proteins that drill holes in these infected cells and kill off those infected cells. And they can also release chemicals, these green dots, that essentially call upon macrophages, and these macrophages basically come next to this infected cell, and once the cell dies off, the macrophage essentially engulfs that cell and breaks it down into different types of things. Now, two other types of cells that T lymphocytes can differentiate into are memory T cells that function in the same way as memory B cells. And we can also form something called suppressor cells or suppressor T cells. And these cells are involved in using a negative feedback mechanism to essentially regulate and tone down our functionality of the immune system. So, our cell-mediated immunity is very effective against cells that have been infected by these different types of pathogens. So these are the major differences between the antibody-mediated immunity and the cell-mediated immunity. But these two different divisions must actually work together to carry out that same function. They cannot exist by themselves. For example, these B lymphocytes need the helper T cells to actually differentiate and produce our antibodies, and the cell-mediated immunity needs these antibodies to actually function properly. And that means these two different systems must work together to carry out that same function.